Okay, the frog. The frog embryo is more complex, and it has a larger yolk. It's a mesolestal egg. It's got a moderate amount of yolk in it, and it affects how the cells divide during the cleavage processes. One thing that's important to realize here is that we've got a yolk distribution that is uneven also. That causes the uneven division of the cells. So with this first cell here, we've got a nucleus at one end and a lot of yolk material down in here. When that first cell division occurs, when we go from being a zygote to the first two-cell stage, it doesn't divide into two equal cells right away. It has a slow process of separation where the two nuclei form here at the end, and a cleavage furrow starts to separate those cells, but all of the yolk nutrient in here slows the process of cell division in the middle. So the nuclei are well divided and the cleavage furrow begins, but we're not getting very far there. The four-cell stage. In the four-cell stage, that cleavage furrow has progressed a little bit further, that initial cleavage furrow, and a new cleavage furrow begins between the different nuclei there at the top, and the process continues. So this gradual movement of the cleavage furrow through that more densely yolked portion of the frog egg. So this is, remember, called the animal pole at this end, and this is vegetal pole over here where all the yolk is. Continued divisions lead to the formation of 8-cell stage, and in the 8-cell stage we begin to see some individual cells kind of populating the top of the embryo that are rather small. Our first cleavage furrow has moved a lot further through there, and our second cleavage furrow is making its way out through there as well, but our third cleavage furrows have isolated those nuclei to the inside, the four cells in here, from nuclei that are out around the margins. So anyway, we have this process continue, and we end up with at our late cleavage stages here. Let's see if I can draw this. Smaller cells at the animal pole, larger cells at the vegetal pole, big block-like cells at this stage of development. Now it's right up in here is where we're going to get the first cavity to form, the blastula, blastocele cavity will develop right in there. So these are different cleavage stages. We're just seeing dividing cells, slowly dividing cells in the vegetal pole, more rapidly dividing cells up in the animal pole. So let's start back over here on the other end and we'll show what goes on with the inside of a blastula. So here inside the blastula stage, we've got smaller cells up here at this end. And we start to see the separation of the cells, leaving a cavity right in here. So here's our blastocele cavity. By the way, you may occasionally run into the term blastomeres. Each cell of a blastula can be referred to as a blastomere, cell of the blastula. These cells continue to divide, and that's even the larger yolk cells down below will continue to divide. So a little later in the blastula stage, still have much larger cells. They're smaller than they were. They're still dividing, these cells out here, but they're much larger block-like cells than those you find at the animal pole. So these are all containing yolk still, and these would not contain so much yolk, these smaller cells across the top here. These cells at the top that have less yolk continue to divide more rapidly and make more and more cells on the top of the cell, and eventually they start to undergo a movement around the outside of the inner yolk cells. So you're going to have kind of a margin here of movement where these cells at the top, the cells with less yolk, the animal pole cells, are moving out around the surface of the inner yolk cells. And that's going to lead to the next stage of development, the gastrula stage. So we're going to set that up in our next couple of drawings here. So here's still a blastocele cavity, yolk cells, animal pole cells up here. So the next thing we see is that these dividing cells, rapidly dividing cells from the animal pole here start to undergo an infolding. At one point, along the margin of the embryo. So these are larger yolk cells down below. Smaller cells from the animal pole dividing here and spreading out over the surface of the embryo. Still have our larger vegetal pole cells here, our smaller animal pole cells here. And right here we're seeing an infolding. It kind of tucks in. There's this moving margin. It's kind of always compare it to like hot fudge going over vanilla ice cream. So you can see where these cells are moving across the surface, and then they just kind of stop and fold in, fold under right there. That's the beginning of our blastopore. The beginning of where we're going to form a gut. Our gut is going to start here and move up and inward through the embryo this direction. So anyway, there's still a cavity. The blastocele cavity is still in here. But right here, we're going to see this infolding of cells form a gut tube that goes through the embryo. And as that occurs, we can change colors and start making our ectoderm in blue. Our endoderm in our orange color. Our mesoderm is going to come from an area just where this tucking in of the ectodermal cells originates, right in here. And so these cells right in here I'll make in red. When these cells first fold in, there's a zone right here that's going to be mesodermal tissue. This mesodermal tissue is going to move out in between the endodermal tissue and the ectodermal tissue here. So I'm going to go ahead and shade in all this ectoderm with this blue, just kind of make a squiggly line to kind of make it. There would be hundreds and hundreds and hundreds of cells that make up this layer around the outside of the embryo. There would be fewer cells in the orange, and I'll just kind of cross-hatch this orange to make sure it doesn't get too confused with the red. So anyway, we have that infolding at the dorsal lip of the blastopore right there. It brings that fold inward, forming the gut, the beginning of the gut. I didn't draw in here our blastocele. You still have a blastocele cavity stuck in this end of the embryo a little bit that we're going to kind of crowd out as the gut grows. So we have ectoderm here. We have what we call the dorsal lip of the blastopore. The beginning of the gut is right here. This is our primitive gut. Remember, it's also known as the archenteron or the gastrocele. Blastocele is still here. Mesoderm in red. Endoderm in orange. Part of the endoderm is going to form the lining around the primitive gut. Part of it is still yolked material, yolked nutrient cells down here. Now another view of the frog embryo. So we have an ectoderm, we have an endoderm forming a gut that goes on through the animal to the other side. And we have mesoderm spreading in between the endoderm and the ectoderm in here. I didn't leave myself much room. Show that mesoderm spreading around there, and it's also spreading around from the ventral lip of the blastopore at the other end. So now we have a layer of mesoderm working in between. It doesn't get there right away, so we'll leave a little bit without the mesoderm in there. But anyway, we've got some mesoderm in between. This is often referred to as the yolk plug stage because yolked endoderm is still seen in a small area where the ectoderm hasn't completely covered it yet. In not all amphibians, but many amphibians, they have a dark pigment to their animal pole when they are first developing. And so that animal pole material, the rapidly dividing cells that forms the ectoderm, will wrap entirely around it, and the yolk portion will be white. And in the yolk plug stage, if you find a bunch of frog eggs, they'll all be this dark color with a little white dot on each of the eggs, and that's where that yolked endoderm is still showing up. It hasn't folded in yet. So on this embryo, the head is going to be way over on this side, so here we're going to have the head developing. we're going to get neural ectoderm, the nervous system, formed right along the very top. The mouth is going to form down here somewhere, and then we'll have the gut going all the way through. A tail is going to need to grow off right here, above there, and all this yolk will eventually be used to help that animal grow and develop the tail and the head and all the structures that would be present there. And the yolk will eventually disappear, the yolk material, and it will just be part of the endoderm in the end. So one more picture to kind of represent what's going on there. We'll have, on this end, we'll have the neural plate ectoderm there, and the beginning of the tail over at the other end. Oh, let's see, our gut tube will stretch all the way through to the mouth. So we'll have a layer of endodermal cells along the gut, and then we'll have endodermal cells along the gut here, and then still some yolk in the gut lining right there. So all that's yolked, or I should say rather, all that is endoderm tissue, forming the gut tube and the portion of the gut tube that still has yolk right there. The mesoderm that grew up through here is going to form our notochord right down the middle of the animal, so there's a notochord developing in here. Just beneath where we're forming our neural plate. So on our surface ectoderm gets really thick right along here, forming our nervous system. So this is all neural plate ectoderm right here. It's going to form a neural tube in the way that we described. The rest of it is simply called surface ectoderm, all this out here. So there's neural tube ectoderm, there's surface ectoderm, there's a notochord that forms through there, there's mesoderm that spreads around laterally. So the next thing we're going to do is we're going to make a cross-sectional view through this embryo to show you what's going on in a cross-section of that. But mesoderm would not only be in your notochord, it would also wrap around the sides of the body and come in eventually in underneath here in this area as well. So all of this would be mesoderm. In this region, and the heart will even form like right in here, in that mesoderm, underneath the pharynx. But if we make a cross-section of this, if we cut through this, like right through here, what you're going to see is you're going to have this neural plate. It's formed right in the middle. Then the rest of the ectoderm around the outside. So we have thin ectoderm around most of the embryo. Where we get to the neural plate, we have this really thick zone of ectoderm. So there's your neural plate ectoderm up there. Our gut tube that formed in there actually has kind of a broad perspective and side view. And let's see. This would all be the yolk stuff down below here. So here's our yolk. What's left of it. Our notochord forms right here. And lateral to the notochord is that lateral mesoderm that kind of folds in here on either side. Eventually it'll go all the way around and meet under the yolk. The left side mesoderm will meet the right side mesoderm beneath the yolk down here. So a cross-section of the body of the developing tadpole would look something like that. Eventually the neural tube will fold in. You'll get a nice tube here at the top, notochord underneath it. Then the gut there with its yolk, mesoderm forming around that here. A body cavity will form within that mesoderm, so we'll get a space to develop out here laterally in the mesoderm. The beginning of the coelom or body cavity in that. So let's label a few things in this drawing before we leave it. There's neural tube ectoderm, should say neural plate at this stage, I guess. Ectoderm is going to form a neural tube. There's notochord mesoderm. There's surface ectoderm here. And that goes all the way around to the neural plate on the other side. The lateral mesoderm, we call this. And then finally there's endoderm. Initially that was the archenteron or primitive gut. We could still call it primitive gut, but we're just going to call it gut. It's going to be forming the whole the digestive tract, pharynx, stomach, intestines, all of that will come from that gut tube there. We already have the yolk labeled. Let's go ahead and label the coelom, the body cavity forming inside that lateral mesoderm that's extending around on both the left and right sides. So this is the basic body plan you're going to see in an amphioxus when you cut through it in an early stage of development. It'll have a neural tube and notochord, it'll have a gut, it'll have lateral mesoderm spreading around and surface ectoderm on the outside of an amphioxus. So an amphioxus wouldn't look a whole lot different from this in a cross-section in its late gastrula stage. And all the other animals that we look at, we'll look at the chicken and mammalian embryos, they're going to have this same kind of arrangement where there will be neural ectoderm forming a neural tube and notochord just beneath that. We'll have layers of mesoderm around a coelom space. There'll be a gut tube in there. So all of it's going to be very much the same as this. Now, you'll recall when we talked about von Baer's law and how the different organisms, they start with basically the same parts: notochord, pharyngeal gill elements, ectoderm, neural tube. All those things are found early in embryonic development, and things get more different as you progress past that. Now, von Baer's law has a little bit of a problem to it in that the method, the mechanism of getting to this stage right here is very different when you compare amphioxus to frog. It's very different when you compare frog to a amniote, like a bird that we'll look at. And it's very different even again to the mammalian condition that we look at. So von Baer's law seems to break down and not really hold in the process of getting to this stage. So it's kind of a misleading law. It's not truly a law if the earliest stages of development are actually more different than this stage of development.